Good afternoon, children. Yesterday we have started with the 6th chapter, that is molecular basis of inheritance, in which we have studied that DNA is made up of, or DNA consists of nitrogenous base, a pentose sugar, a phosphate group. In turn, nitrogenous bases are of two types, purines and pyrimidines. Adenine and guanine are purines, and cytosine and thymine are pyrimidines. Even uracil is the pyrimidine. Okay, then you have studied about DNA, that there is nitrogenous base. Bases are linked to pentose sugar by the glycosidic linkage, which forms the nucleoside, and it also has the phosphate group, which is attached to the 5' OH of a nucleoside through a phosphoester linkage, and as a result a nucleotide is formed. And then the two nucleotides are linked to each other at 3' to 5' phosphodiester linkage, which forms the dinucleotide, and in this way the nucleotides, they form a polynucleotide chain. And a polynucleotide chain has 5' and 3' end free. Okay, that is has a free sugar at its 5' end and a free phosphate at its 3' end. Okay. Then you have studied about the double helix model of DNA, which was given by Watson and Crick. And today we will be starting with the new topic, and that is, the new topic is packaging of DNA. Children, the length of DNA in humans can be calculated as distance between two consecutive, I'm explaining it, right? So now you know that the distance between two consecutive Children, the length of DNA in humans can be calculated. Now you know that distance between two consecutive base pairs is 0.34 nanometer. That is equal to 0.34 into 10 raised to minus 9 meter. Okay, now total number of base pairs in DNA helix in a typical mammalian cell is 6.6 into 10 raised to 9. These are what? Total number of base pairs. We are talking of base pairs. So length of this DNA double helix will be 6.6 into 10 raised to 9 into 0.34 into 10 raised to minus 9 meter. That is equal to 2.2 meter approximately. Okay, thus we see that the length of DNA is far greater than the dimension of a typical nucleus, which is approximately 10 raised to minus 6 meter in diameter. Therefore, there must be some special arrangement to accommodate such a long polymer in a cell of both, that is prokaryotes and eukaryotes. So let us see how the DNA packaging takes place in eukaryotes. We will see first in prokaryotes, then we will see in eukaryotes. Okay, so let us see how it occurs in prokaryotes. In prokaryotes such as E. coli, Escherichia coli, a well-defined nucleus is absent. We have already studied that prokaryotes do not have well-defined nucleus, means they do not have nuclear membrane. So DNA, which is negatively charged, is held together with some proteins and these proteins have positive charge. So this DNA, which is negatively charged, is held in a region with the help of some proteins, and it is known as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. Okay. So negatively charged DNA is held in the nucleoid with the help of the positively charged proteins, and this occurs in prokaryotes where the nucleus is not well defined. Now we will study how the packaging of DNA takes place in eukaryotes. The organization of DNA is much more complex in eukaryotes. Depending upon the abundance of amino acid residues with charge side chains, a protein acquires its charge. Histones are the proteins. Histones are the proteins that are rich in basic amino acids like lysine and arginine. Both of these amino acids carry positive charge on their side chains. So histones are a set of positively charged basic proteins. These histones organize themselves to make a unit of 8 molecules, which is known as histone octamer. Okay, when these histones organize themselves to make a unit of 8 molecule, it is called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer, forming a structure known as nucleosome. Okay, the structure is known as nucleosome. When the histone octamer is surrounded by the DNA, okay, that is the negatively charged DNA, it gets wrapped around the histone octamer, which is positively charged. A new structure is formed, which is known as nucleosome. There are 5 types of histone proteins, that is H1, then H2A, then H2B, H3 and H4. four of these types, that is H2A, H2B, H3 and H4, occur in pairs to produce histone octamer, which is called new body, NU, body. Right, so when this four, H2A, H2B, H3 and H4, they occur in pairs to produce histone octamer, which is called new body or core of nucleosome. DNA connecting the two adjacent nucleosome is called linker DNA and it bears H1. Okay, linker DNA which connects the two adjacent nucleosome, it has H1. I hope this much is clear to everyone. One nucleosome contains approximately 200 base pairs. 200 base pairs are present in one nucleosome of DNA helix. Now, nucleosomes are the repeating units of chromatin, which are thread-like stained, or you can say colored bodies present in nucleus. The nucleosomes in chromatin look like beads on string when observed under an electron microscope. Children, can you see this structure? This is the structure of this single structure is the structure of nucleosome, where you can find the octamer core of histone. Okay, nucleosome, that is, which is formed of histone proteins, right? And the two nucleosome are connected by the linker DNA, which contains H1 unit. Here in the core, there are H2A, H2B, H3 and H4. This 4 occurs in pair. Okay, and so the single is the structure of a nucleosome. Okay, the chromatin is packed to form a solenoid structure of 30 nanometer diameter, and further supercoiling tends to form a loop structure called chromatin fiber and then chromatid. This further coils and condenses at metaphase stage of cell division to form the chromosomes. Okay. Now, non-histone chromosomal proteins, NHC proteins. These are additional sets of proteins that contribute to the packaging of chromatin at a higher level. These regions present in chromatin are euchromatin and heterochromatin. Euchromatin are the regions of chromatin which are loosely packed and stained light in a typical nucleus, and heterochromatin are the regions of chromatin which are densely packed, and when they are stained they are dark in color. Euchromatin is the transcriptionally active chromatin, while heterochromatin is transcriptionally inactive chromatin. Okay. Now, we will be studying about the characteristics of the genetic material. Yesterday we have studied that genetic material is that substance which controls the inheritance of traits from one generation to next generation. Okay, so there are specific properties of genetic material, whether it is DNA or RNA, it should have the following properties, like it should be able to replicate itself. Okay, then it should be stable both chemically and structurally. It should provide the scope for slow changes, that is mutation, which are required for evolution, And the fourth characteristic or the property is it should be able to express itself in the form of Mendelian characters. According to this criteria, both DNA and RNA have the ability to direct their duplications because of the rule of base pairing and complementarity using specific processes, whereas the other molecules in the living system fail to fulfill first criteria itself. That is, they should be able to replicate itself. Like proteins cannot replicate itself. As a result, they are not the genetic material. So then there was the search for genetic material. The discovery of nuclein by mixture and the proposition of principle of inheritance by Mendel were almost at the same time, that is 1869 and 1866 respectively. But the fact that DNA acts as genetic material took a long time to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level, and gradually the question, what molecule acts as a genetic material, got answered. Fine children, this is RNA, where RNA is again made up of nitrogenous base, a pentose sugar which is the ribose sugar, a phosphate group. There are two types of nitrogen bases, that is purines and pyrimidines. Adenine and guanine, like DNA, are the purines, and cytosine and uracil are the pyrimidines. In RNA, instead of thymine, uracil is present. Okay. Types of RNA we will be studying later. Now, we will be studying the transforming principle, as there was the search for genetic material. Okay. Now, Frederick Griffith in the year 1928 carried out a series of experiments with Streptococcus pneumoniae. Streptococcus pneumoniae is a bacterium that causes pneumonia. So he observed that when this bacteria, that is Streptococcus pneumoniae, were grown on a culture plate, some of them produced smooth, shiny colonies, which he named as S-type, whereas the others produced rough colonies, which he named as R-type. This difference in appearance of colonies, that is smooth or rough, is due to the presence or absence of mucus coat on S strains but not on R strains. In his experiments, he first infected two separate groups of mice. The mice that were infected with the S strain died from pneumonia, as S strains are the virulent strains causing pneumonia. So you can see in this diagram, in this second diagram, when the mice were injected by the S strain, that is smooth strain, which were virulent, okay, the mice got infected and the mice died, right? The mice that were infected with the R strain did not develop pneumonia and they lived. R strain, which were non-virulent, when injected into the mice, you can see in this R strain, which is non-virulent, when it was injected in the mice did not die, the mice lived. In the next set of experiments, Griffith killed the bacteria by heating them. The mice that were injected with heat-killed S strain bacteria did not die and the mice lived. Whereas on injecting a mixture of heat-killed S strain and live R strain bacteria, the mice died. Moreover, living S strain bacteria was recovered from the dead mice, S strain hit kill plus R strain living when it was injected in the mice. Okay, the mice died. From all these observations, Griffith concluded that the live R strain bacteria had been transformed by the hit kill S strain bacteria. That is, some transforming principle had transferred from the hit kill S strain, which helped the R strain bacteria to synthesize a smooth polysaccharide coat and thus become virulent. This must be due to the transfer of genetic material. However, he was not able to define the biochemical nature of genetic material from his experiments. So, biochemical characterization of transforming principle was done. Oswald Avery, Colin MacLeod and Maclean McCarthy in the year 1933 to 1944 worked to determine the biochemical nature of transforming principle in Griffith's experiment in an in vitro system, that is not in the living system. In the laboratories they have performed the experiments to know the transforming principle in Griffith's experiment. Prior to this experiment, the genetic material was brought to be was known to be or was thought to be protein. During this experiment, purified biochemicals, that is proteins or DNA, RNA from the hit killed S cells were taken to observe which biomolecules could transform live R cells into S cells. They discovered that DNA alone from heat-killed S-type bacteria caused the transformation of non-virulent R-type bacteria into S-type virulent bacteria. They also discovered that protein digesting enzymes like proteases and RNA digesting enzymes like RNAs did not inhibit this transformation. This proved that the transforming substance was neither protein nor RNA. DNA digesting enzyme, that is DNAs caused inhibition of transformation, which suggests that the DNA caused the transformation. Thus, this scientist came to the conclusion that DNA is the hereditary material. But still, not all biologists were convinced. So, Hershey and Chase performed an experiment. The proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in the year 1952. They carried out some experiments with viruses that infect bacteria called bacteriophages. The genetic material of bacteriophage enters the bacterial cell after the bacteriophage gets attached to the bacteria. The bacterial cells treat the genetic material of the virus like its own genetic material and then produces more virus particles. Hershey and Chase experimented to find out whether it was protein or DNA from the viruses that entered into the bacteria. They took two separate media for growing these bacteriophages. Out of the two, one medium contained radioactive phosphorus. You can see radioactive phosphorus was present in one medium, and the other medium contained radioactive sulfur. So viruses, that is bacteriophage, were then grown on each medium. The virus is grown in the presence of radioactive phosphorus, that is P32. contained radioactive DNA but not radioactive protein. This is because DNA contains phosphorus and not the protein. In the same way, the virus is grown in the medium containing radioactive sulfur, that is S35. Okay, it is difficult to write, children, so please understand. It is S35. Now contained radioactive protein. S35. Okay, so the viruses which are grown in the medium which contain radioactive sulfur, that is S35, contain the radioactive protein and not the radioactive DNA. This is because DNA does not have or DNA does not contain sulfur. This radioactive viruses, that is bacteriophages, were then allowed to attach the bacteria, that is E. coli bacteria, Escherichia coli. As the process of infection with virus continued, the bacteria were agitated in a blender and the viral coats were removed from the bacteria. So the first process or the first step was infection. The bacteriophage, the radioactive bacteriophages were allowed to infect the bacteria, and after the infection they were blended, they were agitated with the help of blender. Okay, when they were spinned in a centrifuge, the virus particles were separated from the bacteria. Right, so dear children, the third step is centrifugation. And when they were spinned in the centrifuge machine, the virus particles were separated from the bacteria. They observed that the bacteria that were infected with viruses contained radioactive DNA, were radioactive, whereas the bacteria that were infected with radioactive proteins were not radioactive. This indicates that only DNA and not the protein entered the bacterial cell from the viruses. Thus, the genetic material that is passed from virus to bacteria is DNA. From the Hershey and Chase experiment, the fact was established that DNA acts as genetic material. But later, studies revealed that in some viruses, example TMV, tobacco mosaic virus, QB bacteriophages, and many more, RNA is the genetic material. Okay. I hope this is clear to everyone. Now, as I told you, RNA was found as one of the as genetic material in some of the viruses. RNA is the ribonucleic acid, and it was the first genetic material to be discovered. It is evident through. Okay, so children, we have started with RNA world. So it is evident through various scientific researches that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. Children, we will be studying about translation and splicing in the later part of this chapter. RNA used to act as genetic material as well as catalyst. But RNA being a catalyst was reactive and hence unstable. right? Was acting as the catalyst. Okay, and as it was catalyst, it was reactive, and when it is reactive, when it reacts with something, it changes. Okay, so it was unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double stranded and having complementary strand further resist changes by evolving a process of repair. The nitrogenous bases in RNA, as I have already explained you, are of two types, that is purines. Examples are adenine and guanine, whereas another type of nitrogenous base is the pyrimidines. Examples are cytosine and uracil. Now children, let us study more about DNA versus RNA. From the Griffith experiment, it was proved that the heat which killed the bacteria did not destroy some of the properties of genetic material. This can be explained by the two complementary strands of DNA, when separated by heating, again come together when favorable conditions are provided. Stability is the main property of the genetic material, as it should not change with the different stages of life cycle, age, or with change in the physiology of an organism. Right? So the genetic material should be stable. In the structure of RNA, the 2' OH group makes RNA labile and easily degradable. At the second position, at the second carbon in RNA, that is ribose sugar, has hydroxyl group, because of which the RNA is labile and it can be easily degraded. RNA is also catalytic, so it is reactive, and additional stability to DNA is also provided by the presence of thymine, that is T, at the place of uracil. RNA have uracil, whereas DNA has thymine. So presence of thymine provides additional stability to DNA. Now both DNA and RNA have the ability to mutate. Since RNA is unstable, it mutates at a faster rate. That is why those viruses which have RNA genome and a shorter lifespan undergoes mutation and thus evolve rapidly. DNA is dependent on RNA for protein synthesis, whereas RNA directly codes for protein synthesis. This proves that both DNA and RNA act as a genetic material, but DNA being more stable is preferred for the storage of genetic information. RNA is better for the transmission of genetic information. Okay, so with this we can say that DNA is more stable and it stores the genetic information, whereas RNA helps in transmission of genetic information. I hope this is clear to everyone. Now we will study about differences between DNA and RNA. The first difference is DNA is double-stranded. With exception of some viruses, whereas RNA is generally single-stranded. Second point of difference is DNA is the genetic material in all living organisms. It is what? Genetic material in all living organisms. Whereas RNA is the genetic material in only viruses. In DNA, the pentose sugar which is present is the deoxyribose. Whereas in RNA, the pentose sugar which is present is the ribose sugar. Now, the fourth point of difference is the nitrogenous bases present in DNA are adenine, guanine, thymine, and cytosine. Whereas the nitrogenous bases present in RNA are adenine is there, then guanine is also there, then cytosine is there, and instead of thymine it is uracil is present. Okay, then DNA is less reactive, chemically and structurally more stable, whereas RNA is more reactive, chemically and structurally less stable. Then DNA usually occurs inside the nucleus and in some cell organelles, whereas very little RNA occurs inside the nucleus. Most of it is found in the cytoplasm. Okay, DNA is found in the nucleus. It occurs in nucleus, whereas RNA occurs in the cytoplasm. Very little occurs in the nucleus, that is RNA. So children will be studying about types of RNA. RNA is of different types like the messenger RNA, which is also known as mRNA, transfer or soluble RNA, which is also called the adapter molecule, that is tRNA, and ribosomal RNA, that is rRNA. Okay, now let us study about the tRNA. Okay, tRNA, which is also called the adapter molecule. The presence of an adapter molecule which could read the code and would bind to specific amino acids during translation was proposed by Francis Crick in 1961. tRNA was known before genetic code and was called sRNA, that is soluble RNA, but later its role as an adapter molecule was reported. Now let us see the structure of tRNA. The tRNA has a secondary structure like clover leaf, you can see on the screen, but its three-dimensional structure depicts it as an inverted L-shaped molecule. tRNA has 5 arms or loops as follows. that is anticodon loop. It has bases complementary to the code. Children, can you see this? This is the anticodon arm or anticodon loop. You can see this much is called the loop, right, which has the complementary bases which are opposite to the codes. Then the amino acid acceptor end, okay, this one, this end, this is amino acid acceptor end. Okay, so at this end amino acids bind. Then T loop, it helps in binding to ribosome. Okay, this is T arm or T loop, you can see, which helps in binding to the ribosome. And the D loop, it helps in binding aminoacyl synthetase, which is an enzyme. Okay, and lastly the variable arm, this small arm, it is what? Variable loop. It is variable in both nucleotide composition and its length. So children, that is all for today. In our next video we will be learning about replication of DNA and further chapter. I hope this is clear to everyone. Thank you.